Hello friends, Ace Botany, next episode. This episode deals with principles of inheritance, second. So I will share the screen so it will be convenient. I think the screen came. So we can proceed. 7 items we'll be discussing in this episode. Of which 7th one will be questions will be given. Theory, entrance key is there. Besides this, characters, emasculation, Blixt, a famous scientist, Monohybrid Cross, then Monohybrid Cross related with it, laws, chromosome number 4 exception. So these are the items we'll be discussing in this episode. So we can proceed. So starting with the characters. So in Mendelism or Mendel's experiment, finally selected 7 characters in Pisum sativa, garden pea. And these 7 characters include stem height, flower color, flower position, pod shape, pod color, seed shape, seed color. And each character has got 2 contrasting traits. So character-wise, 7. Trait-wise, 14. That is that box. After that, you can find a diagrammatic representation belongs to, again, NCERT. There it will be more clear. Exam point of view, they will be regularly asking this area because pod shape, pod color, seed shape, seed color. For students, there will be a confusion. That is why they are putting this question. Pure NCERT. Now, 7 characters, 14 true breeding varieties. Character-wise, when we analyze, for flower, 2 characters will be there. Flower color, flower position. For pod, 2 characters will be there. Pod shape, pod color. For seed, 2 characters will be there. Seed shape, seed color. So all these make 6 characters. Now remaining one is stem height, a little bit more vegetative in nature. That is regarding 7 characters, 14 traits of Mendel in Pisum sativa. Usually one question, theory and runs, etc. I think this page is clear, so we can proceed to second page. In this page, you can find 7 characters of Mendel. In which chromosomes these 7 characters located? And each chromosome has got how many characters? This study was done by a famous scientist, Blixt. And what he observed was 7 characters of Mendel located in 4 chromosomes. Chromosome numbers are 1, 4, 5, 7. And in each chromosome, respectively, how many characters? That will say observe 2, 3, 1, 1. So 1, 4, 5, 7 chromosome numbers, character-wise 2, 3, 1, 1. I think that idea is clear. Scientist name is Blixt, photograph is given there. Now, next idea is that is not directly given in NCERT, but still. Chromosome number 1, which all characters will be located. It is flower color, seed color. Chromosome number 5, pod color. Then chromosome number 4, 3 characters will be there. Flower position, stem height, pod shape. 3 characters, flower position, stem height, and pod shape. And the 7th chromosome, it is concerned with seed shape. I think there's 7 characters, 4 chromosome, each chromosome, which are the, or how many characters will be there? 1, 4, 5, 7, 2, 3, 1, 1. Scientist is Blixt. Maximum is on chromosome number 4 with the 3 characters. There we can find a diagram, this one. So in chromosome number 4, we can find 3 genes. They include, one is concerned with the position of flowers. Second is concerned with the pod shape. Pod means fruit. Third is concerned with the plant height. Pod shape gene and plant height gene comparatively more closely placed. I think this page is clear to all of you, including the contribution of famous scientist Blixt. 4 chromosomes, how many characters each? Now comes famous phylum sativum with the different forms. And what are the phylum sativa we already studied still? It is Phylum sativum garden pea papilionaceous family, pea family. Phylum possesses bisexual flowers. So there can be self-pollination as well as cross-pollination can take place. But in breeding experiment, self-pollination or foreign pollen not acceptable. So in such a bisexual flowers, what scientists will do? There is a procedure called emasculation, you already know it. Removal of stamens or anthers, basically from bisexual flowers of the female parent plant. Emasculation, removal of stamens or anthers, basically from bisexual flowers of the female parent plant. I think this page is clear. Now we can proceed to next one. Now comes Mendel's crosses. First is monohybrid cross. Monohybrid, the term indicates inheritance of single gene. In the experiment part, character taken is height. Height has got two traits. One is tall, one is dwarf. Tall means roughly 6 to 7 feet. Dwarf means 0.75 to 1 feet. And this pure tall and pure dwarf were selected as parents. Left diagram NCERT, right one, it's a representation with the symbols, capital T, crossed with the small t diploid. Parents will produce gamete. Gamete will be haploid, and while representing, gametes will be circled. So capital T, gamete is capital T, small t, gamete is small t. These two will be coupled in F1, and F1 will produce a genotype of capital T, small t. So parents will be usually crossed. Once F1 is produced, first field generation, it will be their own self, same genotype crossed with the same genotype. So capital T, small t crossed with the capital T, small t. We'll be getting two types of gamete from each parent, and these two types of gamete, 2 into 2 will couple, and 4 products will be formed. What are they? Capital T. So in the diagram representation with the symbols, capital T, small t, you can easily identify it. Capital T, capital T, small t, small t, and small t, small t. That is F2. So in 3 cases, we can find capital T. If capital T is there, that is F1. F1 is dominant. Dominant means presence of capital T. So in F2, we can find 3 will be having tallness because at least one capital T will be there. Minimum one capital T will be there. One will be having no capital T at all. Tallness gene is not at all there. So it is homozygous recessive. It will be dwarf. So externally, when we look, we can find 3 tall and 1 dwarf. So ratio become 3:1. That is called phenotypic ratio. And genotypical arrangement of genes, we can find capital T, homozygous, dominant, capital T, small t, small t, not homozygous dominant, still it is dominant. So 3 tall, 1:2, 2:2, then homozygous recessive, small t, small t. So genotypically, 3 types of genotype we can observe, capital T, small t, small t, small t. And the ratio become 1:2, 2:1 genotypically. I think this idea is clear. It's a simple problem type question for theory exam, and even application of it can come in NEET exam. I think page is clear. Now next is, suppose a genotype is given. How we can find how many gametes, types of gametes. So to find the number of gametes, there is a formula 2⁻n. And n stands for number of heterozygous locus or loci. So when we do 1 or 2, it will be easy for you. So genotype, the table is provided. Number of type or types of gamete or gametes. Then which are or what are the gametes? Genotype. So genotype, capital T. From there only one type of gamete can be produced, and that type is capital T. Small t, only one type of gamete is small t. Capital T, small t, 2 types of gamete. So that gametes will be capital T, small t. Now fourth one, capital T, capital R, capital. That is based on dihybrid. Next episode will be clear. Only one type of gamete, capital T, capital R. And last one, capital T, capital R, small r, where we can find 4 types of gamete, capital TR, capital T, small r, small t capital R, small t small r. This is how we can identify number of gametes or types of gametes. And based on this monohybrid cross, Mendel proposed certain generalization, not law, certain generalization. And Mendel's points converted into laws by Carl E. Correns. You'll be remembering rediscovery of Mendel's work in 1900 by three scientists, Correns, Germany, then De Vries, Holland or Netherlands, Treschmark, Austria. So Correns made Mendel's generalization into laws. Now comes Mendel's rule, later become laws based on monohybrid cross. Basically, two laws will be there. One is law of dominance, other is law of segregation. Under law of dominance, certain aspects will be there. Characters are controlled by factors, and these factors are now called as gene. And these factors occurs in pairs in diploid cells. In a situation, capital T, small t. This will be separated as capital T separate, small t separate during gamete formation. And the present world, present situation, law of dominance is not completely acceptable. Little bit we can, but beyond that, many new ideas came, like incomplete dominance, etc. So, law of dominance almost practically not fully valid. Now, law of segregation. What is the meaning of the term segregation? Segregation means separation. How it proceeds? See, two factors in an individual will not blend in F1. Blend means once it is blended, from that we cannot get the parental characters again. This aspect was believed in previously, but now it is becoming obsolete, outdated, blending gone. So when F1 is self, pairs can be recovered. And a haploid gamete carries only one factor. But law of segregation has got universal application. This is only valid law connected with the monohybrid cross in Mendelism. Now, aggregation means combining. Segregation means separation. During the formation of zygote, gametes get aggregated, diploid condition. During the formation of gametes, that individual, the gametes will be separated or segregated, haploid. Now comes locus. Locus means physical position of a gene on a chromosome. That is, place where genes located in a chromosome. And loci is plural. And after that, we have 5 theory question, 5 neat model question with the key. So this is how this episode, we can just rewind it. What all things we learned. First, we explain a box with the characters, 7 characters and 14 true breeding varieties, and what is the exception of plant height. Then character 7. After that, we studied about a famous scientist Blixt, 7 characters located in how many chromosomes? In each chromosome, how many characters? 1457, 2311, and what is exception in chromosome number 4. Then we discussed about Pisum sativa, where terms like emasculation. Then Mendel's monohybrid crosses started. Parents will be crossed. F1 onwards will be self. Genotypic ratio, phenotypic ratio will be 3:1. Genotypic ratio will be 1:2:1. Then how we can identify how many gametes or types of gamete. Then Mendel's law. Mendel not proposed any law initially. Some generalization given. Then Carl Correns developed into laws. Law of dominance. Now it is almost outdated. And second one, law of segregation, only valid law of Mendel. Now what is aggregation? What is segregation? Finally, we discussed about a term, locus. And it's plural, loci. After that, 5 theory question, 5 neat model question with the key. Hoping that this episode you are able to follow. If you like it, subscribe all, like it, share it, comment on it. We can meet later with another episode. So thank you. We can meet later.